I want to thank the organizers for selecting this work to present at the MIDD Immunogenicity Workshop today. I will be going through a case example for a Nivolumab plus Ipilimumab combination in first-line melanoma that illustrates our approach to evaluate the immunogenicity risk and impact across our Nivo and Ipi portfolio. When ADA presents, we know it's important to assess the impact on PK, efficacy, and safety endpoints. Nivolumab is approved as monotherapy and in combination with ipilimumab across many oncology indications. A deeper dive into the impact of nivolumab immunogenicity was prompted when it was observed that Nivolumab ADA increased in the presence of ipilimumab. This slide is a reminder that there was a larger team effort to assess the impact of immunogenicity across nivolumab monotherapy and in combination with ipilimumab and across indications. In the next few slides, I'll present a case example in advanced melanoma that exemplifies this work. CheckMate 067 is a randomized phase 3 trial evaluating Nivo+Ipi combination in a 1:1:1 randomization with Nivo and Ipi monotherapy to assess the efficacy of the combination and Nivo monotherapy versus ipilimumab. The trial was stratified by BRAF mutation status, American Joint Committee on Cancer Staging of Melanoma, and tumor PD-L1 status. Primary endpoints were PFS and OS versus the Ipi monotherapy, and the secondary endpoint was a tumor response rate. As you can see in the top right Kaplan-Meier plot, an OS benefit has been maintained now out to 5 years with the Nivo+Ipi combo in green, followed by Nivo mono and then ipilimumab. At the bottom right are the tumor objective response rates highlighted, showing that over half of the subjects had a tumor response in the combination compared with lower rates for monotherapy. Because we had a randomized trial with the combination therapy and the monotherapies, it made for a complete data set to allow us to examine immunogenicity in more detail. This is a summary of the ADA incidence for Nivo and Ipi monotherapy and in combination from CheckMate 067. Treatment emergent ADAs are highlighted with the pink box, with Nivo mono on the left, Nivo Ipi combo in the middle columns, and Ipi mono on the right. Treatment emergent ADAs are defined as at least one positive ADA sample post-treatment, and for a baseline positive subject, the titer had to increase by 4-fold to be considered post-treatment positive. Treatment emergent ADAs for Nivo in combo with Ipi were 3.5 times higher at 44% incidence compared to 12.3% incidence for the Nivo monotherapy arm. Persistence positive here is defined as 2 or more consecutive ADA positive samples at least 16 weeks apart, and neutralizing ADAs as measured by a cellular-based potency assay. Persistent ADA was low overall, but again somewhat increased for the combination. Interestingly, there was no significant difference in Ipi ADA incidences for the combination compared to the monotherapy. This is a time course of the ADA titers versus time for CheckMate 067 after treatment with Nivo+Ipi combo on the left and Nivo mono on the right. Nivo+Ipi combination is given every 3 weeks for 4 cycles in the induction phase, and this is followed by Nivo maintenance therapy. As you can see, ADA titers are higher during the Nivo+Ipi induction dosing phase on the left compared to Nivo monotherapy on the right, and the decline in titers during the Nivo monotherapy maintenance phase suggests a direct association of ipilimumab dosing and Nivolumab ADA. We next wanted to evaluate the clinical relevance of the ADA immune response. This table shows the rate of acute infusion-related or hypersensitivity reactions for Nivo mono on the left, Nivo+Ipi combo arm in the middle, and Ipi mono arm on the right. Highlighted with the pink box is the total subjects with an event related to the preferred terms listed, such as infusion-related and hypersensitivity reactions. This data showed similar rates across ADA positive and negative subjects in each treatment arm, and this indicates a lack of clinically meaningful association of ADA incidence and these specific events. We next looked at the Nivo ADA impact on PK and tumor objective response rates for the combination arm in CheckMate 067. To the left is a box plot showing the Nivo Cmins at steady state versus ADA status. stratified by titer magnitude using a titer cut of 32. Semen steady states are trending approximately 20% lower for ADA positive subjects. On the top right is a breakdown of the tumor objective response rate by the same ADA titer cut, and it shows no impact on the overall objective response rate. So this was reassuring. However, we also wanted to evaluate the data at the individual level. So we conducted multivariable logistic regression analysis where ADA titer impact on ORR could be assessed after accounting for the impact of other baseline disease factors. The data from this analysis is in the bottom right table. The ADA titers are highlighted with the pink box and showing that there was no significant impact of ADA titer on the odds of achieving a tumor objective response rate, with 95% confidence intervals of the odds ratio that included 1. It's also noted that Nivo after the first dose indicated as Cmin1 in the table, or as an exposure prior to ADA effects, was also not significant in the logistic regression model, and explains why, despite a lower Cmin, objective response rate was not impacted. So it was good to see the lack of an ADA association with proximal effects like hypersensitivity and infusion-related reactions and tumor response. However, we also wanted to assess the longer-term impact using the overall survival endpoint. The Kaplan-Meier OS curves on the left show the combination regimen from CheckMate 067, stratified by the ADA status, again using the same titer 32 cuts. The red and blue curves represent ADA positive subjects at high and low titers, and we ran a log rank test on the OS curves, and the p-value here indicated lack of significance. However, we recognize that baseline disease factors could alter our interpretation. So to account for the differences in disease factors, we performed propensity score matching using 7 baseline disease factors included in the bottom of the slide. And the right plot shows the ADA stratification in this disease-matched data set with more closely aligned OS curves. But you can see that the log rank p-value was even higher, again indicating not significant. One challenge with using propensity score matching is that it shrinks the data set with the potential to reduce the power to detect the difference. So to look at the unmatched data set in a more comprehensive way, multivariable Cox-Proportional hazard analysis was conducted. This slide shows the forest plot from the exposure response OS analysis with the ADA titers highlighted with the pink box. For each titer cut, the 95% confidence intervals included unity, indicating lack of significance after adjusting for baseline disease risk factors. In this forest plot for categorical covariates, the reference denominator is indicated to the right of the colon under the covariate name on the left. And for continuous covariates, the median and the 5th and 95th percentile are indicated. Continuous covariates have 2 hazard ratios defined at the 5th and 95th percentile relative to the median, and a box kind of wrapping these effects, with the blue color indicating that the continuous value is greater than the reference. Nivo and Ipi exposures were not significant predictors of OS, and not unexpected, the M-stage, poor performance score, higher Nivo clearance had increased risk of death, whereas higher baseline body weight had a reduced risk of death. Nivo clearance was previously shown to be a strong predictor of OS in melanoma and other tumor types, independent of the dose and exposure. And this is believed to be associated with a higher catabolic state in cancer patients that present with cachexia, that also then metabolize the therapeutic antibody faster. I've listed several references on this topic at the bottom of the slide. So the totality of the quantitative assessment using multivariate propensity score matching models indicated no ADA impact on efficacy endpoints for the Nivo+Ipi combination in melanoma, despite the higher Nivo ADA. However, the lingering question is, why is Nivo ADA higher when Ipi is on board? Both are checkpoint inhibitors involved in increasing the immune system response. Understanding the answer to this question will be important for developing other checkpoint inhibitor combinations, including the next generation anti-CTLA-4s. As this slide is depicting, mechanistically, Ipi acts primarily in the lymph nodes to improve antigen presentation of tumor antigens. but inevitably non-tumor associated antigens are presented, i.e. Nivo antigens. Ipi has also been shown to deplete and activate Tregs, which are required to maintain peripheral tolerance to self-antigens. Nivo acts primarily in the tumor microenvironment to relieve T-cell exhaustion. This led us to two primary hypotheses as to why Nivo ADA is higher with Ipilimumab. Ipilimumab could lower the threshold for dendritic cell T-cell priming, allowing low-frequency germline sequences from nivolumab to be recognized by T-cells, and/or Ipilimumab could reduce T-regulatory cell activity, reducing immune system surveillance of self-antigens, with Tregs being important for maintaining tolerance. So to address the mechanistic questions, we turn to a mechanistic model. Now, Dr. Chen gave a nice overview of the first published mechanistic model to predict biotherapeutic immunogenicity, and this is referred to as the Chen-Hickling-Vicini model, or CHV model. Her group published the MATLAB code several years ago, and we were able to use that model to make predictions on a phase 1 Fc fusion protein. This was a non-oncology asset we were developing. The CHV model predicted ADA for this phase 1 asset after single dose quite well, but over-predicted ADA after multiple dosing. So we reasoned that T regulatory cells are an important component of tolerance mechanisms. And after multi-dosing of a biotherapeutic, Treg-induced tolerance may be important. As indicated on the left, we added T regulatory cells to the model, and they were included downstream of antigen presentation and were induced in parallel to T effector cells. This work was published in 2019. Inclusion of the Treg cells in the model dampened the humoral immune response, resulting in a predicted ADA incidence after both single and multi-doses that were comparable to the observed data. As summarized in the manuscript, we then went on to predict impact of ADA on PK and PD. The other advantage of having the mechanistic model is to predict ADA under different trial conditions. The phase 2 trial was being assessed with co-administration of corticosteroids as maintenance therapy. So we updated the model further to include the known in vitro effects of corticosteroids on T and B cell proliferation using the corticosteroid dose being administered. And we then predicted a decrease in ADA with corticosteroid use. So using this backdrop of a QSP model that now contains T regulatory cells allowed us to use the model to predict what would happen to Nivolumab ADA if Treg cells were gradually decreased to mimic the action of Ipilimumab. So what is the evidence of CTLA-4 involvement in Treg function? Well, we know CTLA-4 is constitutively expressed on Tregs, and it's important for maintaining peripheral tolerance. CTLA-4 blockade is known to suppress T regulatory cell activity. On the plot to the right are model simulations showing Nivo ADA incidence over time at different percent Treg reductions. The bottom blue curve represents Treg reductions with a predicted ADA incidence similar to that seen in CheckMate 067 for Nivo monotherapy, indicated by the dotted line. The subsequent curves are increasing percent Treg reductions, with the yellow and red curves on the top representing 75 and 100% Treg reduction respectively, with similar predicted ADA incidences to that compared to the observed Nivo ADA from the Nivo+Ipi combination in CheckMate 067. So the model could replicate the hypothesis. So Ipi at a single dose level appears to increase Nivo ADA incidence compared to Nivo monotherapy. But is there an Ipi dose dependence to this effect? This table summarizes the ADA incidence for Nivo monotherapy pooled across multiple tumor types on the left column, the low-dose Ipi combination regimen in RCC and CRC in the middle column, and the high-dose Ipi combination regimen in melanoma on the right. And as you can see, there's a gradual increase in Nivo ADA incidence from 11% to 26% to 38% with Ipilimumab dose. Now, it's true that the tumor type has changed in this comparison, and the Nivo dose is changing. But Nivo ADA incidence is fairly consistent across tumor types as monotherapy, so unlikely the ADA differences is due to tumor type differences. And for the melanoma high-dose Ipi regimen, higher Nivo ADA is unlikely to be due to the lower Nivo dose, since a lower Nivo dose would favor lower ADA. So evidence is accumulating on these Ipilimumab mechanistic effects on T cells that this can increase Nivo ADA. However, can we get to the source of the issue, which is identify the Nivolumab antigenic sequences that get displayed by antigen-presenting cells that then trigger the T cell response? In the upper left box, bioinformatics tools can provide a first insight into potential antigenic sequences that bind to MHC class II with residues that face the T cell receptor. However, these are often followed up using in vitro experiments depicted here on the right top box, where proteins of interest are pulsed with monocyte-derived dendritic cells to allow any peptide antigens to be displayed and the cells lysed to isolate the MHC antigen complexes. The antigens are then identified by LC-MS. In the bottom box are the results of this analysis with nivolumab. A low germline epitope identified in the variable heavy chain between CDR2 and 3 was identified. This potential Nivo antigenic sequence had no protein homologue, no human protein homologue. However, the gene sequence was found in a low germline sequence found in a specific gene that codes for mRNA. Armed with a potential antigenic sequence that was detected in vitro to bind to MHC class II, our next question was, is this Nivo sequence antigenic in vivo? So this is ongoing work to address this. Pre- and post-treatment PBMCs from patients dosed Nivo or Nivo+Ipi in CheckMate 067 will be evaluated to determine if memory T cells are present that proliferate in the presence of suspected nivolumab antigenic sequences. I don't have the results to share today, but I wanted to present this because it feeds into the overall strategy for developing mechanistic models of immunogenicity. QSP models, like all models, require learn and confirm iterative cycles, as illustrated here. For the immunogenicity model, in the upper left here, in silico tools initially make predictions on amino acid sequence antigenicity and use a generic MHC class II population distribution. However, as additional experimental data become available, for example, from MAPS assays or ex vivo T cell recall assays, like we're going to conduct with CheckMate 067, this allows you to refine the antigenic sequence. In addition, if you can genotype the patients for their MHC class II type, you can then use the actual trial distribution of MHC class II genotypes. And that combined with the observed PK and PD and ADA data from the trial, this information can all be fed back into the model to further optimize the prediction. Let me summarize the results here. Nivolumab ADA incidence was increased approximately 3.5-fold when dosed in combination with ipilimumab in the first-line melanoma from CheckMate 067, and this is compared to Nivo monotherapy. However, there was no change in ipilimumab ADA incidence. Nivo ADA was not associated with clinically meaningful increases in hypersensitivity or infusion-related reactions, however, was associated with a 20% lower exposure, but this reduction did not result in an impact on efficacy, as assessed by multivariable analysis and propensity score matching. Mechanistic understanding was enabled using a QSP immunogenicity model. The QSP model, combined with in vitro and in silico bioinformatics sequence tools, allowed for hypothesis testing on the mechanism of ipilimumab-induced nivolumab ADA increases, really for the design of the next generation biotherapeutics. The QSP model predicted nivolumab ADA will increase with T regulatory cell reduction, which was a surrogate for ipilimumab effects. And the future direction here is to assess the T cell recall assays, which are ongoing in CheckMate 067, to see if we can identify memory T cells exposed to predicted nivolumab antigenic sequences to continue to inform this learn and confirm cycle of QSP immunogenicity model development. I'd like to acknowledge the patients and investigators from CheckMate 067 that enabled the data analysis and an interpretation of this work. Also, my fellow colleagues in clinical pharmacology at Bristol Myers Squibb, who are part of a larger effort to assess the impact of immunogenicity across our ipilimumab and nivolumab portfolio. I'd like to thank you for dialing in for the session today, and I look forward to catching up with you in the Q&A session.